So we have specific proteins which are upregulated in response to simulations and others are not. And okay, here is the same data schematically represented. So here's the scheme of the pathway basically. You have the receptors, the Rust level, and then the MAP kinase pathway. And what we indicated here in this scheme, in the leftmost box of each protein, is whether it's induced transcriptionally or not. And you see that most proteins stay yellow, so they are not induced transcriptionally. But we have few exceptions, so sprouty, for example, and the dual specificity phosphatases are strongly induced. So we have actually that some inhibitors of the signaling pathway are induced transcriptionally, and what we also found when we look at the half-life of mRNAs and proteins of these players, we see that the induced ones are very unstable ones, so they can respond very rapidly to transcriptional induction, and the rest, remaining species, are very stable. So we have a separation of the signaling network into a very stable, uninduced part and into an unstable, regulated part. And this we did for many more, or for some more signaling pathway. So for the PI3 kinase signaling, CAMP signaling, TGF-beta signaling, and STAT signaling, and we always found the same principle. So you have induction of very unstable signal inhibitors, while the remaining other proteins are very stable and not induced. So we have negative feedback regulation in the first few hours after simulation as the general principle. Okay, so now the question was, is this induction significant functionally? And this you can test the best way by knocking down responsible transcription factors. So we use this system here, so we transformed red rose cells with oncogenic RAS, looked by microarray which transcription factors are upregulated, and selected 7 of these and made siRNA-mediated perturbations. So this is what you see here. So here's the perturbation axis, and here's the response axis. So we perturbed each of the transcription factor with siRNA and looked at mRNA expression levels, protein expression levels of the transcription factors, and at the signaling activity of ERK and AKT. And what you see here at the bottom is that we have actually, or that transcriptional feedback regulation is significant. So if almost each of the transcription factor actually has an impact, it inhibits, that's the meaning of the dots here, it inhibits basically signaling. There's one exception at the AKT level, but the general principle is negative feedback. And we used, to reduce the complexity of this data, we used a reverse engineering approach proposed by Kolodenko. And so we can reverse engineer the underlying network underlying this data. And you see here that there are several feedback loops predicted from this network. So we didn't go further into this, we tested this network by other means, but I will not go into this in more detail right now. So then the next step was to see whether in a particular cell type we can identify a transcriptional negative feedback regulator by a systems biology approach. So this we did for the TGF-beta signaling cascade in primary mouse hepatocytes. So TGF-beta signaling is initiated by binding of the ligand to its receptor. The receptor phosphorylates the SMAD transcription factors 2 and 3, and they heterotrimerize with translocated to the nucleus, and there induce gene expression mainly involved in antiproliferative responses. And okay, so we did a similar approach as I showed before. So we stimulated cells with TGF-beta and recorded gene expression profiles. And this is here indicated in this pathway map. So we have different time points, 4 hours, 1 hour, 4 hours, 8 hours, 12 hours. We have either regression or induction. And what we see here, very few proteins are upregulated. And we had 2 hot candidates for negative feedback regulation. That's well known, SMAD7. And also another candidate was the SNO oncoprotein. And to investigate this further, we looked at the protein level. And okay, so these are the two hypotheses. So SNO-N might bind to the SMAT factors and SMAT7 might inhibit the receptor level. And to test this, which one is more significant or are they both significant, we looked at the protein levels of the two players with and without TGF-beta stimulation. And what you see is that SMAT7 is basically remaining unchanged, is not induced, while SNO-N is strongly induced. So the hypothesis is that SNO-N is the major feedback regulator in the system. To investigate this in more detail, we implemented, or to try to understand the dynamics of feedback, we implemented a nonlinear ODD model describing receptor activation and SMAT activation, and also the feedback loop. So we have induction of SNO-M, and SNO-M then binds to the SMAT transcription factor and thereby inhibits signaling. And the model was data-based, so we calibrated the model using Western blot data. Here's just an example of total SNO-N expression. We also had SMAT measurements. So here you see the trajectory, the line, and the data points agree well with each other under different conditions. So standard simulation, mixed simulation, and so on. So the model actually worked, but we wanted to test this by an independent way, to test by experiments which were not used to calibrate the model. So we compared SMAT responses in wild-type hepatocytes isolated from wild-type mice with hepatocytes isolated from mice expressing mutant SNO-N devoid of binding to SMAT proteins. And the model predicted, so this is what you see here, what the model predicts are the dashed lines, that phosphorylation of SMAT proteins, so black is the wild type, gray is the mutant SNO-N, should not be changed by depletion of SNO-N. But we should have a strong reduction of SMAT complexes, basically. And so of the transcription factor complexes. And you see that the model prediction agreed well with the data which was then measured. And yeah, we then asked, okay, we have a reduction of SMAT transcription factor complexes in the mutant SNO-N mice. So does this mean gene expression is reduced as well, or does it mean that gene expression is enhanced? And the model predicted that it should be enhanced, and this is actually what we found. So we measured gene expression responses and apoptotic responses as a phenotypic readout and found that both are enhanced in mutant SNO-N mice or in mutant SNO-N hepatocyte compared to wild type